All right. Well,
Yeah,
that's good enough.
yeah, that's pretty good.
This is Audio Munity, a podcast about our body's never-ending fight with the outside world.
Hello, welcome to Audio Immunity. This is episode 54. We're joining you today on recording on April 1st, and I am joined today, as always, by Dr. Kevin Bonham.
Hello, hello.
And I am Matt Woodruff, and I am reaching you today from Emory University. Kevin is at Tufts, so stretching the East Coast, where there is a dramatic temperature difference currently. Here, behind me, you'll see some beautiful leaves. If you're watching on YouTube, it is about 79 degrees outside, sunny. The dogwoods are out, the azaleas are out, the daffodils are up. How's it going in Boston, Kevin?
Actually, it's quite lovely today. It has been cold, but today it's in the high 60s. I looked at I'm not sure if I mirrored or not, but basically I looked at the time course of the temperature this week, and it's basically a 45-degree line going up. I think we're going to peak tomorrow. So yeah, things are lovely. I went outside without a jacket today, and not much is blooming, and all of my bulbs are being devoured by groundhogs or some other creature. Mm-hmm. So that's disappointing. Mm-hmm. But I did find a tiny little, probably half-inch tall hyacinth that managed to survive. So I'm happy about that.
Good. I have to ask, it's warming up. Has the ice been melting in Boston?
Yeah, we're basically snow-free these days. We had a period after the 20-foot snow dump. We had a period where it didn't freeze overnight and it was in the 70s for a week, and basically everything melted. So we've been snow-free for some time now.
Good. No one needs ice in their cities. We are coming into a paper, speaking of things that are cold.
No, we may need to re-record this. This is bad transition.
It's a political statement, Kevin. You just missed it. I used you.
Fair enough.
So we're going to talk about imaging today. Cold things in Matt's life over the last 15 years. Amongst the coldest things was being a grad student in a lab that did an enormous amount of intravital in vivo microscopy. Right?
That's a cool and gruesome technology.
Yeah, really cool, really gruesome. No, it was awesome in that you can take a really cool multiphoton microscope, and I think we've talked about this on the podcast before, and you can actually use that microscope to capture the movement of living cells inside a mouse, inside a lymph node, right? So these are the organs where our immune systems are figuring out how they're going to respond, what antibodies you're going to produce. You can actually see that happen in real time. But in order to see it happen in real time, you need to do a surgery on a mouse that's heavily anesthetized, obviously. You need to be really careful and build up an imaging chamber around the part or the lymph node that you expose. And then you have to sit in a very cold room for hours and hours and hours, capturing movies that will end up playing on somebody's screen for about 15 seconds, but it took you probably 4 hours to produce. The reason it has to be cold is because the microscopes that you use to capture have to be cold, and it has to be dark because those microscopes obviously need it to be very dark. So you end up, as a grad student, in a coat on an 80-degree day in Boston, waiting for these movies to capture over hours and hours. So glad that I don't have to do that anymore as a PI.
One of the joys about being a PI is that you no longer have to sit in cold rooms. I don't know, did you ever how cold was it? Was it 4 degrees? Or
No, nowhere near. It was probably...
This is irrelevant to our discussion, but I definitely, as a graduate student, had to sit in a cold room. Yeah, 4 degrees, and pipette proteins into gels to do biochemistry. And That's miserable because you can't even wear gloves. Or you wear like, nitrile gloves, but you don't have thick warming gloves because you've got to hold your pipette and do all of that. I think that's the most miserable I was in grad school. Totally irrelevant to this paper. There is no biochemistry in this paper, functionally.
No, but hardship around imaging.
Yes,
and
fair.
the reason I bring all that up is because imaging looks cool, is cool, gives you a lot of really interesting information, right? It's the only way to capture spatial information is to image, but it's hard. It's surprisingly hard, right? Doing it well is always challenging, and quantitating it is even more challenging on the back end, right? And we've talked a lot about this on this podcast before, but there is an enormous amount of literature out there that take images that look cool, seem to show the thing that you think that you're showing, and then sort of put them out into the world without really justifying whether what you think you're seeing is actually what you're seeing, right? And the old literature, especially before computational tools and modern image analysis tools, is particularly rife with this, right? Where you'll have a Nature paper that's like, this is true because I saw it, and
showing one representative image of
right, exactly. And a lot of that was sort of the beginnings of imaging, which is just immunohistochemistry, right? Things like H&E staining, or just general histology, right? Even before we get to immunohistochemistry, right? General histology, like basic old school pathology. And so, there's this really interesting sort of linear progression of what we've been able to capture from images, right? And again, why would we do this over something like flow cytometry? And the simple answer is flow cytometry is great if you want to know what a B cell and a T cell look like independently from a surface marker expression perspective. You can't tell if they're in the same place. You can't tell if they're hanging out with DCs. You can't tell if they're in a B cell follicle or in the T cell zone, which are areas within a lymph node that have very different connotations in terms of significance, right?
Right,
So,
and crucially, you're going to be getting, in your flow, you're going to be getting, in the exact same sample, you're going to be getting T cells that are in the T cell zone, and you're going to get T cells that are not in the T cell zone, that are maybe just coming in from the blood through an HEV, or they're getting ready to leave. And in your flow, like, you can maybe draw gates if you know the surface expression that differentiates them, but you're not actually seeing where those cells are. And identifying the markers that are specific for cells in particular places are dependent on you seeing them in those places in an image and knowing what they are. So I think it would be useful. I mean, maybe we should say what the paper is first. So the paper that we're going to discuss, it's not strictly speaking immunology. I think it has a lot of implications for the immune system.
or
But
really,
it's really
in any
about
manner of speaking.
yeah, fair. But it is there's a lot of they label a lot of immune cells in the lamina propria of the gut
They do.
in here. So there's some immunology. Don't give me that crap. But this is really about the technology, an advancement to spatial transcriptomics technology that, again, we've discussed a couple of times. But today I really wanted to sort of go through and talk about the different ways that we capture images, what information we get and don't get from those technologies, and like where this advance is coming from. So the paper is in Nature Microbiology, and it's called Spatial Transcriptomics Maps Host Gut Microbiome Biogeography at High Resolution. And interestingly, all of these authors, I mean, there's not a ton of authors, but they're all from the same place, which is the School of Biomedical Engineering at Cornell. The first author is Ioannis Ntakis. I don't know even what region of Europe these folks are from. And the last authors, I think there's two co-corresponding authors. Is that true? No, There's two first authors. The other is Lena Takayasu, and the last author is Iwigen de Vlaminck. Wow, I should have practiced these before we started recording. But this paper is basically an advanced...
I would add that David McKellar, I believe, is also a co-first author on this paper.
Oh, no kidding. You're right. So there's three co-first authors. Thank you for that.
Yeah, three co-firsts.
And what this paper is it's really an advance in prep of a sample prior to using existing spatial omics technology that allows you to capture both host and microbial transcripts, as well as other kinds of RNA. So we'll get into the details in a sec. But first, I thought it would be useful to just talk about like what are the various kinds of imaging that we can do that give us information about a tissue. Starting with H&E staining, which is these like pink and purple images that, for me anyway, whenever I would see these in graduate school, someone would show an image and I would just immediately, like, my brain would just shut off. And I'm like, I'm just going to believe whatever you tell me because I have no idea what I'm looking at. These
Do you
are...
remember when I asked Uli about those?
No, not really.
It was like second day of grad school, right? And so we were getting the like shotgun talks that you get as a grad student. Like, here are the people that are doing stuff in your department. And I remember Uli, who basically helped bring the lymph node intravital microscopy, technology that I would eventually use into my PhD. He was one of the first to publish on that. So he had a strong background in imaging, and he showed us, I think it was subcapsular sinus macrophages at the time, and he was doing a bunch of H&E staining, and he was just like going through, and I remember it was just like shotgun slides and stuff. And I remember at the end he finished, and I raised my hand and I was like, are we gonna like be taught at some point about how to look at all of the stuff that you just showed us? Because I believe you, but I have no idea what you're showing. And he was like, I mean, you just kind of figure it out as you go. It's like, yeah, cool.
Yeah. Well, I will say that, like, clinical pathologists look at these images all day, every day. You can gain a ton of information because H&E, I forget what the H is. It's like hemolysin or something like that, some kind of toxin, and eosin, which is just one of these chemical stains that was invented probably in the 19th century that binds preferentially to different kinds of tissue. And it basically allows you to see different kinds of cells, different kinds of tissues, and their distribution in a section of tissue. And if you're trained, you can glean a lot of information about a particular tissue from an H&E stain. You can see morphological changes. You can see like the differences in density matter in terms of like, is this a tumor or not? If you're looking at a gut section, you can sort of see villi. And so it's very valuable. I can learn nothing from it, but it matters. It sort of gives you a rough sense of tissue organization and some information about what cells are present. But of course, a lot of cells will stain the same with H&E. And so a lot of techniques have been devised then to get more specific. So one of them is immunohistochemistry, where you take an antibody that is bound to something that provides an additional visual signal, often a fluorophore, but there's other things that you can do. There's people that have done, like, you can attach like different kinds of metal beads and then you can see them on electron micrographs. There's like a bunch of cool things you can do with IHC, but basically this allows you to bind to particular molecules, often proteins, but not always, that will stain certain parts of a cell. So for example, if you were looking at T cells, you could stain like the T cell receptor, or you could stain CD4 with one antibody bound to one fluorophore and CD8 with a different antibody bound to a different fluorophore. And you can see the membrane distribution, you can see the spatial distribution of those particular proteins.
Yeah. And just taking one step back, we mentioned H&E stains specifically. That is among the most commonly used of the chemical stains that pathologists will routinely roll out. There are other similar chemical means that target different things. So in the same way that we can get sort of a diversity in these chemical stains, we can get even more specific by particularly targeting an antibody against the specific protein or molecule that you are interested, right? So we've gained a level of specificity. we've also gained a level of sensitivity, because when you've got that sort of antibody attached to a fluor now, right, you're not looking at the cell level anymore. You're looking at really the individual molecule level.
Yeah.
So now what you're looking at is, what is the upper limit of my magnification capability?
Mm-hmm.
You mentioned electron microscopy. If you take these things and bind them to differently labeled or differently sized gold particles, you can get down to the point where you're looking at an individual gold particle of a specific size, which tells you that one molecule is being bound at that site,
Mm-hmm.
right? So by making that more specific, we have gained both signal to noise and target specificity.
Mm-hmm. And higher resolution. Yeah,
Higher
absolutely. If you have the microscope to do it. You can also do fancy tricks like with fluorophores. You can do something called fret, where you have two different fluorophores, where one transfers energy to another. And so you can see if two proteins are close to each other. But I think we don't want to necessarily go into all those details. But just to say that we can get more specific by using antibodies to target. The limitation there now is based on basically how many fluorophores can you stack for your optics. And often you have a problem where, your detectors are not sensitive enough to split fluorophores that are emitting at wavelengths that are too different. So you have some upper limit of basically ability to distinguish between fluorescence peaks, similar to in FACS, although you're even more limited because not that I understand the optics at all, but I have the impression that it is much harder to deconvolute the signal from an image than from a point in the FACS, in the flow channel.
So that is true, sort of. I would say that imaging capabilities have progressed pretty substantially, even since we were grad students. There are a lot of tricks that you can do now to really boost that. Don't get me wrong, like flow has moved into the spectral deconvolution phase. I've not seen that yet effectively implemented on an image by image basis. I think you theoretically could potentially, but you can now press 6 to 8 colors into an image. The one interesting component of that too is you can't visualize more than 3 colors at a time. Right.
Functionally.
You can't. And people try all the time, right? Like people will take magenta, so it'll be like green, red, blue, and magenta.
But then the overlap causes you problem.
Yeah, but then you have no idea whether your magenta is actually a red-blue overlap, right? Like, so you can't ever visualize in the same way, that you can sort of visualize the data with flow, which is a data analysis problem more than it is a fundamental limitation of technology.
Well, it's not even an analysis problem; it's more a display problem, right?
Yeah,
You're right. It matters. It matters a lot if I'm looking at a paper and I can see the difference versus I have to trust some scatter plot or bar plot or whatever. And this is another reason, we didn't really talk about this before, but this is another reason why imaging matters. Like, I remember in my PhD paper, like I do all this biochemistry to show this localization of this thing, which was thought to be on plasma membranes, and we were saying it's on endosomes. And we did all this biochemistry, we did a bunch of genetics, but then, like, I have a figure in my paper that's like a macrophage. We stained the plasma membrane with red, and then we stained our particular molecule's localization domain. And you can see the plasma membrane is bright yellow, meaning there's co-localization of the outer membrane. And then there's this beautiful circle that's just green. So it's like our molecule is on this endosome and it's on the plasma membrane. That's the whole paper. And I think that one image is probably more convincing than the rest of the paper combined, right? So there's a reason, that not just for the spatial organization, but also just because visually, as we're trying to interpret data, if we can see a picture of a thing, it is even more convincing than if we're staring at data.
Yep, because we're all human beings.
Yeah.
Right.
So this is an aside. I may cut this, but have you seen Ed Yong's book, An Immense World? it's about like animal senses and like all of these different, like we talk about
Oh,
like
yes, yes,
vision
yes.
and smell and hearing, but he also talks about like electroreception and echolocation. It's a very, very cool book. There's also a kid's version that I just recently finished with Isaiah. Um, I'm just bringing it up because like humans, we have this, like, we are very, very oriented around vision. Although he talks about like there are blind people that can echolocate, and which is like wild. Anyway,
Yeah, that's what we need to do. More data display tools based on echolocation.
yeah, I
Yep.
agree. That would be fantastic. Um, so immunohistochemistry. We can stain specific proteins if we have the antibodies for them, but we have some upper limit in terms of how many different colors we can stain in the same tissue at the same time. That's based on the fluorophores. So more recently, I mean, this is skipping a lot of stuff. There's a lot of sort of other techniques that are linked to immunohistochemistry. Of course, you can also express fluorescently labeled proteins if you're in vitro. So there's all of that. But then we come to wanting to know what cells are actually expressing, right? So we have this really great sequencing technology. We've got things like qPCR, where we can look in bulk at what cells are expressing. But if we want to see what cells are expressing in a tissue context, a method that was developed many years ago now is called FISH, fluorescence in situ hybridization, which is essentially the same idea as immunohistochemistry, but instead of using antibodies to guide your fluorophore to the right place, you instead have nucleic acid probes that will then bind through complementarity to their target. So
To a gene product, right? So when you say express, what we're talking about is
Exactly.
the kinds of mRNA signals that the cell is using to produce protein,
Exactly.
right? So you're moving away from protein targets and towards nucleic acid targets now.
Exactly right. So you have transcripts now of specific genes, and you can. The nice thing about this is that developing an antibody, like, there's lots of techniques and people are very good at it now, but even still, like, it's not always easy to get an antibody to the target you care about. You got to like go inject a rabbit or a mouse with your thing. You got to use the right adjuvants. You got to purify all this stuff. With nucleic acid probe, it's quite easy to just like, I have this gene sequence. I have this probe sequence. It's not exactly perfect, but it's very, very easy to target a particular probe to a particular molecule. Downside of FISH, of course, is similar to the downside of immunohistochemistry in that you have a limited number of targets that you can actually identify. So what we're going to talk about today is a kind of sequencing that is basically sequencing in place on an image in small little areas. But there's another kind of spatial omics that Matt is much more familiar with that is essentially like FISH, except that the probes, rather than being single fluorophore probes, are essentially barcodes, where you have multiple fluorophores attached to a given probe. I think you have multiple versions of the same probe, right? So you can sort of see the same transcript multiple times with different fluorescent barcodes, and that allows you to target many, many more transcripts. So you can get something closer to looking at the full transcriptome. It's still not the full. What are you up
Well,
to?
you
Like
can
a few
actually.
thousand?
Well, so there are full transcriptome products out there, but it's
For
yeah.
barcode based?
Yeah, but
Okay.
here's there are some caveats to this. So let's dive a little deeper into the spatial transcriptomics. So what Kevin is talking about is two fundamentally different approaches to this technology. One approach is based on sort of a traditional, what we call now a traditional single cell RNA sequencing kind of approach, right? And so in single cell RNA sequencing, what you do is you take a cell and you put it in this little emulsion, so one cell per emulsion, and in each emulsion you have a unique
By emulsion, you just mean a bubble,
bubble. And in each unique
a
bubble,
liquid bubble, maybe.
yep, each unique bubble, you have one cell, and you also have a unique barcode. That barcode, so basically what you do is you pop the cell inside the bubble, you lyse the cell, and that barcode integrates itself into all mRNA in that bubble. right?
This is a nucleic acid barcode now.
This is a nucleic acid barcode. And then what you're doing basically is you're taking that newly barcoded information, you're combining it across all of your cells, you're sequencing all of it. And then what you say is, okay, well, I've got 50 genes here that came out of my entire sequencing run. 35 of those genes all had the same DNA barcode at the front. And what that tells you is that those 35 genes came from one specific bubble, which means that they were from one specific
Those 35
cell.
reads specifically, right?
Yeah, exactly. 35 reads. Yeah, easier to conceptualize as genes, which is why I use. But yes, and you could also say like these other 15 that are left to the side, they all have the same barcode, but it's different than the first one. So these must have come from a different cell, but they're all grouped together, right?
And
so,
just to be clear here, the barcode is a sequence of DNA, a specific sequence of DNA that's known up front. And they probably do some filtering to make sure that it's not going to be found in the genome of the thing that you're targeting, right? Or in the transcriptome, at least.
Correct.
So it's a unique sort of, it's like essentially random, but
yes,
it shouldn't align to, it's the barcode.
Yes.
Because it doesn't align to anything else. And also probably its location also, in the transcript.
Yeah, it's positionally aligned correctly, right? So that is how traditional single cell sequencing works, okay? Now imagine you take that bubble technology and you transfer it. Now instead of your bubbles being in solution and having a cell incorporate into those bubbles, imagine you took a physical slide and you put little tiny dots all over that slide. And each one of those little tiny dots contained one of those unique barcodes. And you did exactly the same thing. So now when you put tissue on top of that slide, parts of the tissue contact all of these different tiny dots, right? And so the slide itself allows you to transfer those barcodes to whatever cells are in that physical location on the slide. So it's actually really the same technology. It's just that you are spatially embedding that unique DNA barcode.
And crucially, you know ahead of time which barcode is in which location on the plate or whatever it is.
Exactly.
So that when you deconvolute at the end, you can overlay specific transcripts at specific locations on the slide.
Exactly. Now, here's the rub on that technology. The rub is that you can only resolve down to the density or the size of the unique barcode that you printed, right? And even if you did that like infinitely small, right, you would always have boundary problems. A B cell and a T cell are interfacing, right? And they're not going to line up nicely into one side of the barcode or another. You're never going to get cellular resolution because that's simply not how the barcoding technology works. What you get are these micro regions functionally decided by where you placed those barcodes or where the company placed those barcodes on the slide, right? So this is called spatial transcriptomics, the idea that you could derive the transcriptome through this sequencing-based technology and then sort of back calculate where on the slide those transcripts were coming from, right? And this was the earliest version of this technology, and you can build it all the way out to full transcriptome sequencing because it's just a sequencing reaction, right?
So your limitation there is the number of reads you sequence.
Yes.
Very, very rare reads are less likely to be sequenced. Mm-hmm. So in theory, if you could sequence infinitely, you could find every transcript. In reality, as with any technology like this, things that are rare are harder to find, and so you may not find enough of them or enough of them to be confident that they're there, depending on what your sequencing depth is.
Right. So then we move into a more recent or more modern version of this technology, which instead of being based on the barcodes or being based on really any sequencing technology at all, is going to move towards a DNA probe-based approach, much closer to FISH, like you were just talking about. So for each gene that you're looking to assess, you have the hybridizing probe part, and then you have this tail, and this tail is basically designed so that it encodes a unique chain of fluorescent molecule attachment sites, right? And
It's a
so
different kind
basically,
of barcode.
different kind of barcode, right? And so you're right, you'd have, let's say, 5 probes for CD4, right? So they would hybridize at different places on the CD4 transcript, but all of those would have the same fluorescent barcode to basically help you assign the identity of that molecule. And so basically what happens is you go through this multi-step reaction where you go through, on our system, 16 different cycles where you add in, on cycle 2, a new set of DNA slash fluorescent probes. And basically what you're doing with your microscope is you're capturing at each pixel whether on that cycle it flashes red, yellow, blue, or green, right, at each cycle. And so basically what you're doing is you're capturing fluorescence flashes at each spot over the course of the experiment, right? And at the end of all of that, you say, oh, well, CD4 was encoded by blue, green, blank, 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 red, blank, blank, yellow, right? And so everything is integrated. That code comes back and you say pixel 800,045 flashed that correct CD4 fluorescent signature. Here's the trick with that.
You literally type that out with your keyboard.
That's exactly right.
Pixel 1?
You do it all by hand. So what you can do then is you can get a sense of what the most likely transcript type is at each pixel location. So now your resolution once again falls back on the resolution of the microscope. How many pixels can you realistically assess within your experiments to get you a good approximation of the tissue, which works pretty well for the most part, but you have an additional constraint. The constraint there is that you have to pre-design all of those probes. You have to know exactly what you're looking for. It is not agnostic the way that normal sequencing is agnostic, right? Anything can come up on a normal sequencing run. Here, you have to previously have identified which probes you're going to use because you have to put a DNA barcode on all of them. And the transcript density per cell is capped at the number of pixels that cell is made of, right? You can't have more than one transcript per pixel. So you're actually creating a max transcript density or a max transcript expression per cell, which is fine, except if you go in and try to ask how, like, if you want to know the full transcriptome of a cell, you physically can't, right? Let's say that a cell is made up of 10,000 transcripts or 10,000 pixels, which would be an enormous cell, right? You're still only capturing 1/3 of the total human genome if each individual transcript is present exactly
only once, yeah.
Which is not what happens. What happens is you're going to get a bunch of housekeeping genes in there, a bunch of actin, a bunch of like all this stuff. And you can equilibrate at some level, but it turns out, you know, within this technology, there's a real give and take, right? You can go for breadth of transcript coverage, but the broader you go, the more likely it is that you're actually going to miss the interesting stuff that you're looking for, right?
Yeah.
So you have these two technologies. They're good at different things. One is fully sequencing based, but you're never going to get to cellular resolution. The other, you can get to cellular resolution, but you really should target into the biology that you're looking for. It is an ongoing, I wouldn't say tension, but it's an ongoing discussion as to, you know, where these different things are utilized effectively. But in this paper, we are going to be talking about the first, right? Being able to use broad sequencing technology to not only investigate the context of human and mouse transcripts, but also microbial transcripts.
Right. So the major limitation of that sequencing based thing, aside from the spot nature of it, the resolution, the area level resolution, is that the way that we find the things that we're going to sequence traditionally has been basically to target poly A tails. So in eukaryotic cells, when you make an mRNA, you transcribe it in the nucleus. This is coming back to central dogma of molecular biology. You transcribe it in the nucleus. That RNA gets processed, and before it moves out into the cytoplasm to potentially get translated, one of the things that happens is you add what's called the polyadenylate tail. So you add a bunch of A's onto the 3' end of that transcript. There's also a 5' cap. These things aid in transcription and regulation, a whole bunch of stuff. But crucially, not every RNA product in a cell is polyadenylated. Messenger RNAs that code for proteins are generally polyadenylated, but there are a bunch of other small RNAs, or even long, like link RNAs, long non-coding RNAs, microRNAs, snow RNAs, I can't even remember, small nucleolar, maybe? Anyway, there's a bunch of other RNA products in our cells that don't have poly A tails, and bacteria do not polyadenylate their transcripts, right? They're oftentimes transcription and translation are happening at the same time in microbes because there's no nucleus, their genome is just out there in the cytoplasm. You start transcribing an RNA, a ribosome finds it and starts translating like basically immediately. So the regulation is very different, but what that means is that traditionally when you do spatial omics in a tissue like the gut, where you've got lots of microbes, these spatial technologies, the microbes are essentially invisible. We'll see in this paper they're not completely invisible always, but they're generally invisible. So the innovation in this paper is essentially to just say, rather than trying to come up with some like fancy way to like target all of the mRNAs with different probes or something like that, they take like really like this entire paper boils down to one additional step, which is that prior to the normal library prep, they do an enzymatic reaction that just adds a poly A tail to basically every transcript, or every RNA. Just any RNA, you find an RNA, you add a bunch of A's to it. Like that's honestly, like that's really just all it is. I mean, I say that, but this is, I'm sure, a ton of work, and the validation is very good, I think.
I imagine it's not actually. I imagine discovering it was a ton of work, but sure. I imagine like the actual doing of this is actually not all that complicated.
A lot of work is all of the validation,
yes, exactly.
And controls that they likely had to go through. There was probably a huge amount of troubleshooting.
Yep, I imagine that's all true.
Yeah. So, but basically that's it. So now, once we add a poly A tail to all of the RNA in the cell, or all of the RNA in the tissue rather, now they just go through the normal process to grab onto poly A tails, do the spot-based sequencing that Matt described earlier, and then the analysis now you have the entire universe of RNA in the cell, and that's what they describe in this paper. So I think Figure 1 is actually like it's a purely, not purely, but it is primarily a description figure. Like it's essentially a cartoon of the process. I found it incredibly helpful, sort of laying out the process here. So we don't have to go through all the details, but one of the things they do is they describe two different versions of this spot-based sequencing that have different properties in terms of their resolution. So the Visium, which is one that they spend a lot of the time on, their diagram basically shows that each individual spot is 55 microns across in diameter, and the distance between the center of individual spots, or it looks like the difference between the centers, but oh yeah, that does work. Okay, so 55 micron diameter spot, and then the center of each spot is 100 microns separated. Comparing to the StereoSeq version, where each spot is 0.22 microns across, and the distance between spots is 0.5 microns. So it's like a 10-fold higher resolution, maybe 20-fold higher resolution. So each spot contains a much, much smaller area, which means you can get a lot more discrimination. And I'm not sure what the primary difference is in terms of like if you were trying to decide. Obviously, higher resolution is better. Is StereoSeek much more expensive, or
I'll bet it's less expensive based on everything I know about
oh,
10x and my
okay.
intense dislike.
Fair enough. 10x is the company that produces the Visium. Uh, just to be clear on that.
No, I mean,
It may be,
it's good.
it may
It's
be
good
that doing the
technology.
chemistry.
Honestly, they sue the crap out of everybody. Their business model is to sue everybody in the space, and
Cool.
I don't appreciate that. So
Awesome.
Yep.
With StereoSeek, like it may be that doing the chemistry in those small spots is hard, so maybe the error rate is higher. I have no idea. Just totally speculating there. But basically, they have these two technologies. They show an example of a tissue where they can overlay the spots and color by the individual transcripts. This technique, by the way, of adding the poly-A is, they call it PAP, and PAP, and then they essentially can just like layer on the different transcripts. Like once you analyze the sequencing data, you can say like, here are where my microbial transcripts are, here are where my eukaryotic transcripts are, here's where my microRNAs are, and you can visualize these in a bunch of different ways that they show in the rest of the paper.
Can I ask a really dumb question
Sure.
here? So you've got the tissue,
Mm-hmm.
and you've got the lumen,
Mm-hmm.
and the lumen has your microbial compartments in it.
Mm-hmm.
But lumen is empty space.
It's not.
I don't understand what we are looking at here.
I mean,
Is every cross section here across
Fecal
a piece
pellet.
of poop? Okay.
yeah. Yes, almost certainly.
Just making sure that, so like every time we're seeing microbial colonies here, we are cross-sectioning across poop.
I mean, not exactly, because like up in the ileum, for example, like up in the small intestine, you don't have necessarily like very well-formed pellets, right? Like in the colon, like you absolutely would. But like, you know this, when you look at lymph nodes, like
Just making sure that
Lumen-
because like it's not
it's
really
lots of
lumen.
fluid.
Right?
It is
No,
lumen.
I understand.
There's fluid, there's microbes in the
I mean, but
the
you're not
lumen.
imaging the fluid, you're imaging the poop.
I'm pretty sure that most of these are cross sections of fecal pellets. Yes.
Okay.
I don't know that for
just
sure. I
making
didn't actually see that. I was assuming that was true. Yeah.
That has been my assumption too, because I don't otherwise know where we would get these transcripts from.
I mean, you can in principle take a section that contains fluid that is still there, and microbes are in the fluid of the lumen of the intestine. They're not only in like, but yes.
Okay.
This is probably poop. Anyway, so that actually brings up a good point. So what they're doing here, they're looking at the intestine, and in Figure 2, they sort of show this throughout. They have a very nice little diagram of a long mouse intestine, and they are taking different parts of that. So if you imagine like the intestine as a sausage, essentially, right? Especially where there's a fecal pellet, it's a stuffed sausage, and you take a cross section where you get little discs, right? Of the intestine and the lumen. So around the outer ring have the epithelial barrier as well as the lamina propria, which is the section behind the epithelial layer. And then in the center, you have where the microbes tend to be, although we'll see it's not a perfect separation there. And they take sections from different parts along the intestine. They do now they're comparing regular Visium to Visium with the polyadenylation, with the PAP. And helpfully, every time they have PAP, they have this little like Pac-Man looking thing that I assume is meant to be an enzyme. I don't know why that keeps showing up there, but then they show basically if you look at the bacterial molecules in a given sample, with the normal Visium across all of the different sections, they have no microbial molecules, more or less. In the Visium plus PAP, they have very few in the small intestine and the ileum, but lots in the cecum and in the colon. And then they show a comparison basically between bacterial taxa and host genes. They show, again, for each individual spot, they're sort of quantifying how many of host versus, how many host mRNAs compared to other kinds of RNAs, like ribosomal RNAs, RNAs that are unspliced. So a step that I skipped in my central dogma explanation is that for eukaryotic genes, they're transcribed with introns. Those introns need to be spliced out. That happens again in the nucleus prior to polyadenylation. So you see that basically in regular Visium, you have almost no unspliced transcripts. With the PAP, you get lots. And here by lots, I mean like 10 or 15%. And then you also get a small percentage of microRNAs, which are these tiny little RNAs that are involved in gene regulation of various kinds. They show, in E, they're showing basically where microbes are, and it's exactly what you would expect. It's in the poop. It's in the poop. It's in the lumen of the intestine.
It's not.
You don't think poop is lumen?
No, it's in the luminal space. It's just not the lumen.
Okay. Right.
So here's the thing. I believe everything in this paper. It's just that what we're investigating here is the progression of bacterial taxa as poop is formed and descends across the digestive tract. Right now we have immune components that can be tracked sort of along the way. But I just think the framing of it is funny, right? It's like I think it's framed in a funny way.
Okay, fair enough.
Yep, which gets more funny because they don't really directly say like here's what we're actually assessing here, which is just poop colonies, and that's okay. It's okay. That's what you study. It's a good paper made into Nature Microbes.
Yeah. Yeah, like how often do you think they should have used the word poop?
I don't know, so many times.
Let me actually Control F. Do they say feces at all in this paper? I'm searching for feces. Phrase not found. Fecal. Phrase not found. Poop. I'm guessing. Phrase not found. Nope.
We would only have to.
Might as well check. I'm being complete here. Okay.
Okay, so we're 6. We live with 6-year-olds. And,
well, Isaiah's 7 now.
Yeah, well, okay. Caroline's about to be 7.
All right, so F, they start talking about, so when you get a microbial transcript, you can then align it to database, and you can identify what species this is from. Actually, they don't get species level resolution. They're looking at microbial families. Honestly, like the details of this aren't super important other than to say, like, these are all the microbial families you expect to be in mouse poop. Maybe some interesting differences between different sections of the intestine. You can't do this in people. You typically don't get microbiome samples from the ileum, for example, in people, unless you're doing like ileal biopsies and stuff. But we can see here that like certain bugs are much more present in the small intestine and ileum. Other more common stool microbes are in the cecum and the colon. Again, this is all expected. And so then what they do, and again, like, I don't know how super valuable this is, but they sort of show you, like, with one of these sausage sections, part of it is the lumen, and they label that as one zone. And then they can sort of label other zones based on, like, how deep into the fecal pellet in the luminal space things are, and they can say like where are these bugs spatially compared to the epithelial barrier. And perhaps interestingly, some of them tend to cluster right around the epithelium. Some of them tend to be more towards the center of the section. And all of this is maybe interesting from a microbiome standpoint. All of it's maybe interesting from a host pathogen perspective. Bugs that are going to promote inflammation are probably going to get closer to the epithelial barrier. So there's maybe some of that. Honestly, like none of this jumped out at me as being super critical in this, but it's basically, it's sort of a, not a control exactly, but it's showing the potential value here where we can see and quantify the differences between where microbes are located. And so you could imagine that if we looked in an IBD setting or we looked in a colon cancer setting, as they do later in the paper, we could see, are there differences under different conditions in terms of where microbes are, which might be a function of immune response. It might be a function of expression of antimicrobial peptides. It might be a function of things like mucus and mucus degrading microbes. So all of those things might matter. And then H, I think this is, to me, this was sort of one of the more interesting places where they're basically showing, if you look with the Visium, you don't see any microbes anywhere. This is, again, expected. But also there are a couple of these important non-coding RNAs that you can see little hints of in the Visium, but you can see them really densely in when you add this PAP. And you can sort of see spatially the microbes are more in the center sections and the host transcripts are out in the edges. So that's cool. They also, they highlight, oh, this transcript, this makes sense because blah, blah, blah. I highlighted those, but I skimmed over it because it's not my field. And I don't think that's really the point of this paper. The point of this paper is really showing that we can discriminate here between different areas. We can see the differences between cells in the crypt, which is the sort of base of a particular villus, microvillus, versus the ones at the top. You have different kinds of regulation happening at various stages. So in the epithelium, there are stem cells in the crypts down in the base, and the goblet cells that produce the mucus, those are in the base. And then as cells develop, they from the stem cells, they sort of migrate up to the tips of the villus, and then they get shed off from the tips of the villus. And so there's lots of interesting cell biology and tissue biology here that is potentially visible when you can also see all of those regulatory RNAs, along with the typical protein encoding transcripts. Yes?
Yeah.
Okay.
Yeah.
Matt's just going to nod along.
No, yes, I buy all of this. This is they're clearly using this technology effectively to be able to amplify the bacterial signatures that they're getting, and in combination with, and we'll see this in the next figure, but in combination with the StereoSeq technology, I think that we're getting pretty good resolution here. I think I agree with you. I think that the value here is the method. And I would say off to the side, this is the kind of thing that, people have approached me about in the past, because we do have a spatial transcriptomics machine in the other room. And this is something I was this is actually too hard. I don't think that you will be able to design a probe set that would allow you to sort of get at this, and they have put together a method here that really has overcome a lot of those limitations. So this is not an easy question. They've put together a reasonable solution.
Yeah, and so as Matt said, figure 3 is using this more high resolution technology. We're essentially seeing the same thing, but at much higher resolution, where we can actually see sort of walking along the villus. And it might be actually worth talking about the scale of these things. So I mentioned that the Visium is 55 micron diameter sections separated by 100 microns. And in this image, I'm no pathologist, but they show that basically the length of an individual microvillus is 150 microns. So with the Visium technology, we're talking about really 2 spots, maybe 2 to 3 spots total across the entire length of a villus. And again, we're not perfectly aligned, so we're probably going to overlap some lamina propria. We're probably going to overlap some lumen or pellet. Whereas with the StereoSeq, here we're talking about 5 micron between centers. So for a 150 micron villus, now all of a sudden we've got tens or dozens of individual spots. And so here we can really start to separate out the different locations and identify transcripts that are present in various places. So they show some of the things you can see, they can label mature enterocytes that are at the tips of the villus, as opposed to the goblet cells that are sort of more in the crypts, and TA cells. I can't remember what TA cells are. But they're also then labeling a bunch of immune cells, T cells, B cells that are mostly in the lamina propria, right behind the epithelial barrier. So we can get much more resolution, and then you can also zoom in on the individual microbes, and they show various colors, which are basically impossible to discriminate as far as I'm concerned in this image. But in principle, you can discriminate different kinds of microbes at different locations, which again, visually, it's impossible to distinguish, but you can then do a heat map and say, where are the bugs relative to the epithelial barrier here?
I find this figure infuriating.
Okay.
And the reason is because I think that these are beautiful data. I think these are beautiful data. I think, you can't ever, through this technology, assign like true confidence of a transcript, being one thing in one cell versus another. But if you do get to this kind of resolution, you can be fairly confident that the majority of the spots are giving you fair representation. So all of that. And so you do all that work, right? And you do all of these classifiers. And now you've got discrete cell populations, and you spend half a page on a single image with every single color for every cell type, instead of pulling this back, showing, like, you could have done this and then spent a quarter of the page with individual cells zoomed in to one of these villi, right? And shown exactly where all of these cell types line up in the actual structure, and it would be substantially more impactful, I think, than this. And this, this shows you the complexity of what they're doing in sort of like a zoom out. But I wish that they had visualized this just a little bit differently, because
they do in the supplement, right?
At some level, yeah.
I think that's in supplementary figure. What is it, like 8?
Yeah. Yes.
They do a little bit more of that. I think the value here, because like, if you look at this with an H&E staining, you know where the different kind of cells are, right? So you don't really need the transcripts. I think the value here is that you can sort of see in the scope of this entire thing, you can see how the transcripts are aligning, and that like the transcript level identities are aligning with where, because like, mature enterocytes are going to be at the tips of villi. Where goblet cells are. Like, this is basic intestinal biology. Like, if you zoom in so close and you really focus on individual things, you're going to get bashed by the pathologists. So they're going to say, like, that's not where a goblet cell should be. But
as it stands, I don't know where an immature enterocyte versus a dendritic cell is. I don't know where my endocrine cells fall on this. Just, you run into that issue that I was talking about before, which is that there are limitations in our own visual capacity to pull in information. And there are some easy ways to get around that in this kind of setting. And I agree with you. There's work in the supplement that's a little more clear, but I wish we had incorporated some of those tools into this main figure.
It is a very, very large image that they pull out there, but they're trying to display an entire cross section of an intestine. So
I got that.
I don't know. I give them a little bit more credit. Although the black spots that correspond to, like, I'm not sure which bug those are supposed to be there in the center. Exactly. It doesn't matter. Anyway, it doesn't matter. It's a cool paper overall. So then they go, the second half of this figure, the other half of the page, is sort of split into host versus microbe again. They're showing a bunch of these non-transcript, non-messenger RNAs that they're finding in the host. So they do this kind of interesting thing where they show spliced transcripts versus unspliced transcripts. And they do this analysis, which I've never seen before, but basically they're saying, like, which genes are 5 standard deviations off of 1-to-1 ratio of transcript to non. So this is basically saying, like, are there transcripts that are where you have more of the non-spliced versions relative to spliced versions? I don't actually know the significance of this. They highlight one in particular. This I can't even see on my screen here. Got to zoom in, the CDK8, which is a tumor suppressor gene. And what they say is that often tumors will regulate the splicing of transcripts. So it's interesting that we have a bunch more unspliced copies of this tumor suppressor. And this is going to lead into figure 4, where they look at a tumor model. But it wasn't clear to me in the narrative of the paper, like, why they pulled this out, other than to say, like, oh, well, but we should look at cancer. But anyway, it doesn't matter. Then they also look again at all of the different microbial families now. Here they're looking at genera, comparing the richness of the bacteria, the number of individual unique molecules, bacteria-associated molecules, and they're showing where they live in relationship to the epithelial barrier. Some microbes they find are more closely associated with the epithelium. They get closer to the epithelial barrier, and some are more in the center, as I said earlier. Again, like the specific details of this to me were not super like revelatory, other than to say that they can do this analysis, which is kind of interesting.
Yeah, and you kind of skipped over D and E, which is actually what I was hoping that they would do earlier, which is like so they have this sort of cool heat map where they're looking at gene expression by distance from the outer edge in D. I think you could have done this actually with cells themselves, like how many cells from the edge to this, and show color coding as in E in a single villus, like where are these cells setting themselves up? Can we identify all of those things? But anyway, you can actually see that IgA band, for example, which I think is a really interesting thing, right? You can visualize exactly where the IgA producing ASCs are in comparison to the rest, really, of the immune compartment. And I think that that's some interesting insight that gets pulled out of that because of this analysis. So I actually really like the way that they sort of incorporate the spatial information into D&E.
Yeah, I guess I sort of skipped over it mostly because like that doesn't feel specific to this technology. Like this you could get with normal spatial omics, right?
You could, but I think it's showing the interplay between the mammalian and the bacteria, right? Like showing that you can do kind of the traditional cell-based versus, anyway.
Yeah. Okay. I buy that.
I like it. I
buy that.
Generally like.
But they also do this thing, you mentioned colonies. This was something that was sort of surprising to me. So they do this autocorrelation analysis where they say, like, for a given species transcript, how likely is that transcript to be near a transcript from the same species, right? So if microbes are like randomly distributed throughout they're sort of like well mixed in the pellet, you would see low autocorrelation. But if they're forming colonies, if you're getting bunches of microbes, then you can see that they would have higher autocorrelation. And so they find like certain bugs are more likely to form larger colonies inside this fecal pellet, which I thought was kind of interesting. I would have liked to see if there's any autocorrelation, like if you see colony formation associated with the epithelium. Most like commensal bugs aren't gonna like directly associate with the epithelium, but I thought it would have been interesting to see, like, are there dynamics in terms of distance from the epithelium that affect the colony size? But that's just sort of a random question there. Okay, so that's figure 3. So, and then figure 4, as I mentioned, now they're going to go into a tumor model. So again, like, you want to show disease relevance for just about everything. They highlighted that one tumor suppressor gene earlier, and they use that as a justification, like, hey, we should look at a tumor model. And they do a couple of interesting analyses where they're looking at a region of the intestine where there's I think it's just a CRC, right? Colorectal cancer model. Oh no, this is Ilium section. It's a APC min positive mouse. So I don't know that model, and I didn't read into it. But this is spontaneous tumor generation. You have a section of Ilium where there is a tumor, and now they're going to use the same analysis. And so in A, they show what one of these cross sections looks like, and then they start doing some analysis looking at bacteria versus host cells. And B is this. I actually really liked the way this was displayed. So they have it's a density plot of bacteria versus host cells, and they have a line where the boundary is for the epithelial layer, and then they sort of highlight the section that is tumor, and then they show like where are the microbes relative to where are the microbes in the host cells. And basically what you can see sort of is that at the tumor site there are a lot more microbes that are like right up against the boundary than there are in the normal tissue section. And then they break this down now by individual microbial taxa at the genus level, and you can see that some microbes are actually infiltrating into the tumor. So Clostridium is getting into the tissue in the normal tissue, but where there's cancer, Clostridium sort of has this hard cutoff, and it's not getting into the tissue where there's a tumor. So presumably that has to do with inflammation, or it has to do with the immune system or something. I have no idea. Other bugs, there's some difference between the tumor and the normal tissue, but it's not nearly as clear, the differences. And then they basically do the same analysis, but now for immune cells. So you can see that, for example, you've got lots of dendritic cells and macrophages and T cells infiltrating into the tumor, getting into that area of the tissue, but not like B cells, for example. And a bunch of the epithelial cells are obviously not found in the tumor. It's just the I can't remember what cell type, what the etiology of the tumor cell type is. Yeah, you can see the infiltration of the immune cells. So that's kind of neat that you can sort of compare the microbes and the host cells. Again, that's the sort of like promise of this technology. And that's the whole paper. We just got a little immunology there at the end.
Yeah.
And it's cool. I don't know. Yeah, I really liked it. I thought it's a neat technology.
It is.
I'm excited to think about, like, I feel like there's a lot of ways you could apply this in like human colorectal tissue samples, in IBD. And the fact that you don't need a specific probe set, you don't have to know what you're targeting. You can do this in sort of a more discovery basis, I think is pretty cool.
You know what it brings up for me is, like, there's been a couple instances where we have talked about whether we believed microbial signatures coming from specific tissues that are supposed to be sterile, like the brain, for example, like placental development, where there have been papers out there that have basically said, hey, like, actually we do see. And everyone's like, no, but is it contamination, right? Like, which makes total sense. I think a technology like this could actually clear some of those things up, because if you found that those signatures were there and also they localized to a very specific location in a growing placenta, I think it would actually go a long way. Like you were saying before, when you see a thing, it can sometimes clarify exactly what we're talking about, rather than doing general sequencing. So
yeah, I like that idea. I suspect that the purveyors of those theories would say that the biomass is too low, and so when you have a sequencing base, because like a lot of those contamination studies, they're not doing metagenomes, they're doing amplicon-based sequencing.
Yeah,
you're only seeing it because you're doing the 16S amplification first, and yeah. But I totally agree with you. If somebody showed me this kind of thing, and they showed that, like, actually we see these colonies in this location and it's consistent, yeah, that would be more convincing.
Yeah.
I don't expect we'll ever see that, but that would be very convincing.
There you go. There's our
Yeah.
scientific snark for the day.
Fair enough. It's not the only one that we've had.
No, it's true. We've had a couple. Most of it me.
Yeah, so I think this is cool technology. I'm interested to see if we can apply this in more disease contexts. Obviously, I'm sure that these people are working on that. I'm sure they have a bunch of collaborators that are clamoring for these folks to do those kinds of analyses. But I think it's super cool.
Yeah, agreed.
All right. Well, this has been Audio Immunity, recorded on April 1st. This entire episode has not been an April Fool's joke. And thankfully, I have not seen some of the over-the-top April Fool's jokes that I feel like have proliferated in previous years. Maybe it's just because I haven't been on social media today, but...
Could be. I think it's because the world is sadder.
Yeah, that's true. I did get one from a web database company that I use called Base Row, or that I used, and they're like, we call this analog Base Row, and it's just a file cabinet, and they do a bunch of marketing around the file cabinet. It was dumb. I didn't like it, but maybe it's just because the world is sadder. Anyway, we are now primarily on Substack. You can find us at audioimmunity.substack.com, audioimmunity.org, AUDIOUNITY.org is still there. It now points to the Substack primarily, although archived episodes are there. Substack is nice because I can just upload the video and it does all the transcript stuff. It makes the little clips for sharing on social media. I can post in one place and it gets distributed everywhere rather than having to upload to a bunch of different places. So it's great. If you would like to follow us on Substack, you can do that. We're posting everything for free. If you would like to support us, you can get a subscription. It's by month, and the baseline is $5 a month. But if you want to pay less than that, just send us an email, contact@audioimmunity.org. I will give you a discount code that you can use forever at whatever level you want to support us at. We would really appreciate it. The music at the beginning and the end is composed by Rachel Reinick, and I don't think there's anything else. We'll see you next time.
Peace.
Sometimes